similar profiles of individual fatty acid species in different lipid classes. Another thing that I really like is that you can work all the way from 10 microliter up to 100 microliter with the same protocol, no change in volumes of solvents or buffer. It's still the same protocol, 300 microliter of the BUME and the second solvent and the buffer, and you get great linear recoveries of the lipids of most polar and most lipophilic, and the range in between. So next step was to move into tissues. And basically we just tried to adapt the same protocol, the same proportions of solvents and buffers, and do it in a semi-automated way. And that way is it's a generic method we have for all kind of tissue biomarkers, regardless of the metabolites, lipids, proteins, or something else. So the starting point is these 2 milliliter homogenization tubes that are filled with beads. You can buy them from Precellis, Bertin, and there are a lot of providers nowadays. So we collect from 15 to 150 milligram tissues, keep them deep frozen, and these racks, as you see, are compatible with the 96-well format. So we can process 24 samples, still using the robot, if you like, to dispense and pipette extracts, and keep the samples cold in this rack. Frozen tissue, 500 microliter into these tubes, do the homogenization using Precellis instrumentation. That is just a mixer, a very powerful mixer. And then the addition of heptane, acetyl and acetic acid. You do this manually, so it's more manual work. Mix all the samples, and you can do these 24 plus 24 samples in a time, so it's 48 samples together. Simply collect the upper phase, pull the extracts, and then you have a lipid extract in a couple of hours for these 48 samples. We tried heart, brain, liver, colon, and muscle. And just to have homogeneous samples to start with, we used powdered dog liver as a matrix to compare the lipids with BUME and FOLH. And as you can see, we get really nice data, again, for the acidic polar phospholipids, better recoveries for BUME, and great linearity from 15 to 150 milligram sample. Then we did a study with real tissue, not only the powder, because powder is not the true challenge. So it's heart, split in half, and used with the BUME or FOLH method. And again, great data, comparable. And we also looked into the lipid species, and they look the same. So to conclude, we have now developed a novel method for total lipid extraction, the BUME method. And it's not only fast, simple, and reproducible, it's also chloroform-free, ideal for automation, linear, compatible with high fat content, and showing highly comparable and also blind diet. Acknowledgement to my colleagues that has been part of the work. It's Gun-Britt, Ralf, and Sinikka, and Sahlgrenska, Jan and Maria. So thank you for your attention.
Any questions for last? Yeah, please.
Any questions?
Okay.
So this is lies. Would you provide some comment about how about the water-soluble parts with these methods? Because for the Fokker methods, we can simultaneously extract the hydrophobic and hydrophilic parts. And this one only get the lipids? So this methods?
Yes.
So for the chloroform methods,
yes.
we get the hydrophobic and hydrophilic parts.
Yes, you mean the aqueous phase and the lipid phase.
Yeah. So for this one, you get the lipid phase, but how about the water phase? You don't have the water phase.
Yeah, the water phase is equally volume as the lipid phase, and you can do everything with that as well. You can do organic acids or whatever, amino acids and stuff, so you can use that. And if you like, you could actually, after collecting the lipid extract, you could add chloroform to flick the aqueous phase to the top mix it quickly, centrifugate, and then you have automated aspiration of that aqueous phase as well, if you like.
Yeah, same direction. Only the acidification of the water phase, I doubt a little bit about the stability of the compounds in the water phase with adding acetic acid.
The stability of the polar metabolites
Yeah.
in the aqueous phase in the presence of acetic acid we haven't studied. Maybe that's something you want to do if I provide you with all the details and we can do something together. But we have
So
looked
you
at
have no, so the efficiency of the lipid phase is dramatically reduced when you don't use the acetic acid, or?
the acetic acid has no major impact actually. We during the optimization work we tried different concentrations and there are small differences with and without. It's better for fatty acids, for example, that I didn't show here. So for the acidic lipids it's better, but for the others it doesn't matter. We looked into some lipids, acid sensitive lipids, the what are they called? It's in here. Those who have aldehydes in a lipid. This is anyway, and they were stable. Sorry for not remembering right now. So it doesn't seem to hurt the lipids at least. Okay.
Yeah, I have a question.
Yeah.
So you show is you add 300 microliter your solvent to extract lipid. So I'm wondering what is your requirement for fluorine tissue? What is that volume? Fluorine tissue, right? When you add, you give 300 microliter your solvent, right? What is your fluorine tissue, the volume, the mass?
The mass, we work everything between 15 milligrams to 150 milligrams works with the tube to homogenize.
Okay.
If that's what you ask.
How about biofluid?
From 10 to 100 microliters in the 96 well format with the same protocol. Microliters of sample.
So to find out your mass spec, which mass spec you use to measure lipid?
Like I do sample preparations. It's Marcus, my colleague, who do that.
Okay. Thanks.
Yeah, please.
So
yeah.
it was, I think it's a science instrument that has been used for some of the work. I can get back to you with details after the meeting, if you like. It's well described in the publication. There is there is a link here to the journal where the method is published for biofluids. So that's very well described what methods was actually used for the lipids.
Okay, a simple question. I'm very interested in your work. It's very interesting, When I attend a meeting here, my students in my lab are also doing the lipids extraction with the FUSH methods. And when I am a PhD student, I also use the FUSH method to extract the lipids. So my question is, I have noticed that in terms of the serum samples, that the recovery between your methods and the FUSH methods are almost the same.
Yeah.
So I don't know how much about that machine, the robots. I mean, in our small lab, if this kind of method can be used, we don't do large scale samples with lipidomics, but we need to do the lipids extraction with the FUSH method.
Yes.
So I just want to know if this kind of method be used in small lab. Okay.
I'm sure it works very well to do extractions, but so you end up with more work per sample, but it's still probably easier by having the upper phase to do the work. And I'm sure you have mixers, separate mixers, so you can do all that without, you don't have to pipette mix, you can go to a mixer instead. So it will work for sure, using the same volumes and amounts, I'm sure.
Okay, I'm afraid you have to move on.
Yeah.
Let's thank the speaker again.
Thank you very much.
The next speaker is actually my co-chair, Dr. P.K. Vaguna. He's from the Sri Ramachandra University in India. Apparently, it's a change in the topic. Oh, it's not this one, yeah, okay. It's relevance of machine learning approach in classifying metabolites associated in asthma and COPD. Yeah, please, yeah, thanks.
It's not closing. File is not closing, I don't know. This is not my file. Understand? No, it's not working. Good morning to all. I'm Dr. P.K. Raghunath from Sri Ramachandra University. 15 to 20 millions of patients has got recorded in India alone. And then other information, this one give you. Then regarding the COPD, the case which we won't compare this one, chronic obstructive pulmonary disease, preventable and treatable disease with some significant extra pulmonary effect that may contribute to the severity individual. To my knowledge, it looks very simple on the definition. Even I don't know how they are using preventable, but most of the time, I heard that it's very, very difficult to. When I said that COPD, it is almost, they said somehow they should go for surgery. The very complicated issues are being addressed there. Otherwise, in the definition they use, I don't know why the preventable. I don't know, probably they thought that preventable, the keyword can be used with many of the disease. That's what. Otherwise, it is very deadly disease, if I'm correct, when compared to asthma. That means very well that asthma is very well responds to all type of the treatment. Whatever you go for inhaler, all those things. Even if you're not with any steroids, are there. There are so many things are there. But COPD, ultimately, they go for only the surgery. That's where we thought this one. And then, what are the conditions that contribute to COPD? So generally, the mucus hypersecretion with the enlargement of tracheobronchial submucosal glands and disproportionate increase of mucous acne. In addition to that, inflammation of bronchoalveolar mucus metaplasia and hyperplasia, also is being reported in COPD. Moreover, respiratory bronchitis is a critically important early lesion. As you know, the lesion, the keyword generally we always use when you go for cancer, other deadly diseases. That's what they are trying to use this here. And apart from that, ultimately it distracts, all the alveolar wall. Ultimately, the patient is not able to recover. Then compared to this one, asthma, it is very deadly. That's what I heard. Then apart from that, what are the complex, this one, asthma and chronic obstructive disease or complex conditions with the imprecise definition. That's what still the physicians are facing a lot of issues in clearly defining COPD. Apart from that, there's another word, asthma, COPD overlapping syndrome. That's where we tried a small piece of information. Due to time constraint, I'll rush through. As you know very well that, the metabolomics involves the quantitative measurement of time-related multi-parametric metabolic response of living systems to pathophysiological stimuli or change in this. That's what, Davis and Bernard, they gave a very nice definition, which generally I used to love it. Okay. That's why I use the definition. And then what are the things we used to do in metabolomics? Generally, we always go for efficient and unbiased separation analytics, as I said. Even, the last speaker has given a wonderful presentation on how to play with the lipids. That's where this one. And then apart from that, in second stage, we always go for detection. And third, identification, quantification. These are the major areas in which we play, as a the latest entrant or the latest developments is taking place on these key areas only. Generally, this concept we tried in our linear regression a simple method. Then the equals in regression, one variable is considered independent or variable and other one is dependent. That's what we always identify, dependent and non-variable, and finally we go for the relationship analysis. And then there's another small piece of analysis we use, PCA, principal component analysis. This PCA we always generally basically is a multivariate analysis based on projection methods. Basically it's being used extensively in generally, and this we can always extract and display the variation in the data by using this PCA. And PCA is a combination of original data. Then each successive PC explains the maximum amount of variation possible not accounted by the previous PCS. That's the issue. Then generally PCs are orthogonal to each other. These are the general interaction we thought of giving on this PCA. Then the third step, initially this was the material methods. We started we tried initially before we go and play on the wet lab. We thought we should have a scheme at least SAB in silico. That's what we try. Use text from the what medical literature is available. Then from there we try to this principal component, this above, this one. From the the left hand side, we're able to identify only the top principal component which had eigenvalue of greater than 1 were selected, not other things I'm sure we left out. These are the scatter plot of PCI and the combined linear regression side and PCI2 this regular side. We were able to combine and finally we were able to come out with this kind of scatter plot. It's very clearly listed clearly. Then finally, in the multilayer perceptron model to classify the metabolites associated with the regression PD, I use the PCA and finally we end up with we were able to identify 90% about this this one for this one. And finally this is the work this one, and finally even this guy is also able to identify the PCA, and finally this is best the model we done. And finally this is the multilayer perceptron model to classify metabolites associated with the Osman COP based on the feature extent. That's on the clearly we're going to demark. It's very clear that all of them on the line. This is a ROC plot of where generally we used to plot the sensitivity versus specificity. If you're going to cross, I'm sure all the best statistician model. We can go and play on the other side this one. Then insight from this is what we got from this one. The multilayer perceptron model to classify metabolites associated with the Osman COPD based on feature extraction by PCA showed greater than 90% in comparison to a model based on feature selection. But still, it's very near to this one only. Then what is the scope? The similar metabolites classifying models can be built using radial basis function, which we have not tried. Probably we may use in the future. Its efficiency can be compared with the current model also. A model classifying metabolites associated with the comorbidity condition can be attempted. That's where we are trying to, link comorbidity. That means connecting both the symptoms in the same case. And essentially, I like to, I'm sure, finally, I like to acknowledge the which given beautiful, more number of metabolic list from its literature, and from this my team and the hospital. This is the thing. Ultimate aim is of this work. We like to try. Ultimately, we are planning to go back and test it on the wet lab side. Probably that's what we are aiming for. To ultimately, we want to track this asthma COPD overlapping syndrome, which accounts for more than 1% in my hospital. That's what we are trying. Thank you, sir. Thank you.
Okay, thank you. Okay. Question? Okay. Thank you.
Very nice work
Yes, thanks.
and amount of work
Yeah,
also. In machine learning, my impression is that you make a model
Yes.
either for comorbidity or other things which are closely related or not closely related. So you use what happens that I
Actually, we can always increase the number of we try to go for only 45. I think, sorry, I remember 75 this one. We'll try to add more number of descriptors. And then finally, we can improve the efficiency of the model. That's what we may try in the near future. That's what we are trying. That's what we are saying.
okay.
More number of cases, more number of descriptors will definitely will improve the what you call prediction level. That's what we try. Sir.
And your curve look different. That is sensitivity.
Yes, so this is specifically the sense. This one, no, that's just mission the left or right hand side. I'm sure it's there. It's efficient only. That's what.
Okay. And last question is that this was for morbidity.
Yes, sir.
But if we are following 2 or 3 or multiple diseases at different times,
Yes, sir.
one is for progression, one is for regression, then have you made any model for maybe 3,
No,
4, 5?
actually, to my knowledge, this model, though we try to compare asthma and COPD, it goes more rather than put from the vet lab or a physician side. Probably we may end up with a nice at present preliminary work which is being carried out. And my basically I was a medical setup. So we thought of releasing this. So, thank you.
From majority, just use a simple PCA for their classification. The asthma and the cough, they separated by classification.
No, basically, when we say metabolism, we always use increase or decrease. Not simply, we're not identifying. When I say metabolites, either we always get the value from the vet lab, either they say that it increasing. Apart from the, I'm sure there's another area wherein they identify, unique things. But here in model, we go for either increase. In some metabolites, it's always increase. Some metabolites on the COPD decrease. That's where we tried for these two parameters, decrease and metabolite.
That's nice.
Yeah, that's right.
So since asthma and cough, they have low definition, separate them. So even though they have something
something.
can you use these parameters as a guideline to redefine which group they are asthma, which group are cough?
Yes. That meant ultimate. Our next. That's what is given a scope. Probably we may develop a software based on this model. We may, I'm sure, in case somebody is going to come out with a new metabolite or whatever it is, my software may predict whether asthma or COPD area is emerging. There's a new entity is emerging called asthma COPD overlapping syndrome itself. So there's a lot of confusion is taking place in my system. That's what this one. Thank you. Yes, sir. So many, that's what a metabolite as well as genome wide. That's what somebody was given a beautiful explanation yesterday. Unless until we link these two, genome wide and the metabolite wide, probably we'll have better understanding. That's what we. Yeah, sure. That's what show. So one level further. Yeah, definitely. We need more insight on this. Yes, sir. Thank you, sir.
Okay.
Thank you very much for.
Okay. Yep. Let's thank Professor Laguna again. We shall move on to the next talk, and it will be given by Dr. Nigobam Sanjit Mehte from Premier Biosoft in the USA. He's going to tell us about this software tool called SimMat as a bioinformatics tool.
Good morning, everybody. First of all, I would like to thank the organizer for giving me this opportunity to present our work. We are a bioinformatics company, and we have a number of bioinformatics software tools that caters to the demand of mass spectrometry-based omics research. Popular software tools consist of SimGlycan, the only software tool for glycan identification and quantitation. SimLipid, one of the most popular lipid identification software. MultiVision for multi-imaging, mass spectrometry data imaging, and ProteinIQ, that's the downstream data analysis for protein quantitation and statistical analysis. And finally, we come up with this SimMat, which is a high throughput software tool for metabolite identification and further statistical analysis. So definitely, I'm going to talk about the software workflow, which will assist identification as well as downstream analysis that may consist of statistical analysis and, of course, in future, pathway and networking analysis for metabolites. Needless to say, there have been many presentations on the analytical platform for metabolite profiling. So one of the most common analytical platform for metabolite profiling is liquid chromatography mass spectrometry. Is quite consensus. Before beginning of this presentations conference, I used to have many points and why liquid chromatography is and others, but is more suitable for metabolite profiling between NMR and LCMS. So the answer is already there and have been covered by many of the presenters. So I just include high sensitivity and requirement for low sample amounts. So this is one plus point for liquid chromatography mass spectrometry workflow. There are many advantages provided by LCMS workflows that includes number of peaks detected by the workflow, and then its ability to distinguish isomers on the basis of another dimension, that is retention time too. And in addition to this retention time, there are other aspects which can distinguish metabolites on the basis of exact mass search, isotopic, this one, pattern matching, and another is tandem mass spectrometry data analysis. So it provides numerous advantages, and one main challenge with this workflow is that it provides and it generates huge data sets, which is very cumbersome and which is at times very tedious to analyze. And any of the statistical analysis, identification, pathway analysis, those are downstream analysis. The first challenge we have to face is this data reduction. Typically in metabolomics analysis, there are multiple biological replicates as well as technical replicates that need to be studied just to ensure that data is valid, the quality is controlled, and there is no confounding factor while you try to identify or characterize a particular metabolite. One of the main challenges provided by this workflow is data, huge data sets. First one, very frankly, to provide metabolomics data analysis, there are many software tools existing in some of which are commercially available and some of which are freeware, which try to address metabolomics challenge, but none of those software tools provides a comprehensive or single platform solutions for data analysis. So by single platform software solution, I mean this LCMS data processing will require peak picking, peak detection, and of course other feature identification and retention time alignment. And then metabolite identification that may include database search techniques, and on the basis of MSMS, if there is tandem mass spectrometry workflow involved, then definitely there will be another software tool or another workflow that will require interpretation of MSMS spectra for confident identification of metabolites. Performing statistical analysis for identifying differential metabolites. Many of the presenters have included PCA, PLS data analysis, and PLSDA, and other even analysis of variance. That is quite simple. That's very effective statistical technique. So those statistical techniques are also required. And then finally, pathway analysis for the identified metabolites, which will bridge this informatics and the biology. So these are the steps. Software tools, they provide comprehensive solutions, which on which a particular a researcher can perform all these steps simultaneously. So this is the main motivation for us to develop SIMMET software, and up to a certain extent we have achieved. That is 1 to 4 we have achieved in the first version of SIMMET, and in the upcoming versions of the SIMMET software we are going to integrate this 5th part, that is pathway data analysis. So this is the schematic representation of how work. So first of all, irrespective of what mass spectrometer instruments, by mass spectrometer instruments I mean vendor specific mass spectrometers. So you can always import raw data into the SIMMET software using their file formats. Like if I am using Bruker's instrument, I may want to import the data using FID file or YEP file or BAP file. And similarly, if I have a AVSciEX or now currently SciEX instrumentation, then I would like to import the data using SciEX WIP file format. So that kind of facility is provided by SIMMET. So once the data is imported into the program, then what you can do is you can go for data pre-processing. That include peak detection and then feature identification, peak alignment, and without having to go to metabolite identification, you can directly go for statistical analysis, or like principal component analysis. Then from principal component analysis, you can identify metabolites which contributes to the separations of samples, okay, using correlation ellipse or Hoteling's T-square ellipses. So from those statistical technique, you can identify only a handful of metabolites which are really affecting the difference in the characteristics of those biological samples. And then you can identify those specific metabolites instead of identifying the whole metabolome. Or another workflow is you can directly take that pick list from the detected pick list, and then you can go through the whole metabolite identification process, which may include exact mass search, isotopic pattern matching, and MSMS database matching. So in the MSMS database matching, there are many different techniques for metabolite identification using MSMS database matching. For lipids and for glycans, we are quite successful using in silico fragment matching. But unfortunately, in small metabolites, small molecule metabolites, which are less than 500 Dalton of monoisotopic masses, that's not very successful. So what we have integrated recently is we have a partnership with NIST MSMS database, and it contains like around 9,000 compounds, and the number of MSMS spectra and the number of this one ions are statistics is available here. So we directly apply a pattern matching algorithm between the observed spectrum and the standard spectra, and we come up with the best possible metabolite structures for a particular spectrum. So that's the identification process mechanism we follow. We can have annotated MSMS spectra, and also you can quantify metabolites, level-free quantitations, and any level methods for quantitation can be used. And finally, all the results can be exported into these report formats, namely Microsoft Excel, HTML, CSV, and other image formats like JPEG or PNG file formats. So, this is the summary of what we have provided. Generation challenges include generation of large number of LC peaks from raw data file, and we provide molecular feature finding algorithm that combines peaks from ADAX, higher charge stats, and other relevant peaks in the mass spectrum. Significant number of peaks corresponding to noise. This is very problematic, and it may sound quite easy, but definitely when we get into the actual raw data file, it becomes difficult to remove noise peaks corresponding to the actual analyte. So what we do is remove noise based on shapes of the LC peaks and data from LCMS runs of blanks, quality control samples, and technical replicates of biological samples. So we provide this technology for, methodology for removing noise from the samples. Missing peaks, we perform gap filling. That means if a particular peak, LC peak is detected in a particular peak list and it's not observed in other peak lists, then we go to the raw data file of other peak lists and see whether any intensity within that LC timeline is observed or not. If it is totally blank, then we leave it as a blank, that is, the peak is not identified. And if there is any peak intensity, if in that particular timeline, if intensity is observed, then we extract that intensity from the raw data and we fill the gap here. So accurate identification of metabolites using MSMS data, I just explained that we use NIST MSMS database, and we use our own proprietary scoring algorithm, which is capable of performing pattern matching as well as retrieving the exact metabolite. So just to validate, verify what our software is capable of, we have acquired coffee samples. The method is explained here. We use Bruker's QQ top instrument, and the LC we used was SPLC U3000 RSLC from Thermo Scientific. So the specifications of this method is explained here. And these are the 13 coffee samples we have used here, Ristretto, Apregio, Roma, and other. For each coffee sample, there are 3 biological samplets, biological replicates, as well as 2 technical replicates. So finally, for each sample, what we have is we have 6 raw data files, and all in all, we have 78 raw files to be aligned and to be analyzed. So this is typical CMAT software user interface, wherein you can import hundreds and thousands of raw files in a single click. And this is another interface wherein user can model the experimental design, like data can be clubbed into biological replicates or technical replicates, and if a particular template is already available on how to perform peak picking, peak detection, or what kind of statistical analysis to be performed once the alignment is done, then he can select it and just click OK, and yes. Okay, another aspect is data normalization. So this is very important. We spend a lot of time on trying to identify which normalization technique is best suitable for metabolite, this one, statistical analysis. But we never could come up with a specific answer. Sometimes at certain point, we always find that, okay, Pareto scaling is very effective for this particular sample, but when I go back to another T sample I have and another mid sample I have, we try to apply Pareto sampling, Pareto scaling, but it's not that effective. It's not able to, give us the normality or the data normality which we expect from the data. So definitely there are different types of, this data normalization and data transformation techniques. Many research papers are also available on which data transformation and which type of normalization should be used at different conditions. But definitely it should be left to the user, because he is the person who should know that, okay, whether a particular normalization technique gives him a appropriate or meaningful data which he is expecting out of the data. So we left it to the user, to be selected that which one would be the most appropriate, data normalizations and, this one, transformation techniques. So very, very quickly, we perform, pick list in batch mode, pick deconvolution, that means picks, in LC pick, if you look into liquid chromatography mass spectrometry, that extractor and chromatogram, a peak may be a representative of multiple isomers. So that deconvolution is required. So different types of deconvolution algorithms are available. Local maxima, second derivative of Savitsky-Golay smoothing technique, and then Mexican head technique, that is second derivative of normal distribution. So again, same case, one algorithm is not effective in all cases. I have many lipid data wherein, all the three algorithms fail to identify PC lipids, and finally what I have to do is I just need to force that, okay, this is the ADAC and this is what feature for this particular peak is, and I have to search database. So we provide 3 algorithms for users to select, and by default we, definitely if the person is not that experienced in this deconvolution, then definitely the program will guide and the program will select which one will be the most appropriate deconvolution technique for its data. So data reductions, this is very important. So this is one of the most important aspects of accuracy of the statistical analysis you are going to perform as a downstream. So for this sample data I am using, when I subject this data into MGMine and XCMS, they give me around 75,000 rows. So 75,000 rows, that means if someone, I think some of you may be familiar with Metabolomics too. So Metabolomics has a capacity to incorporate 5,000 rows for further statistical analysis. And 75,000, it consists of many redundant peaks. That means at least one metabolite may have at least around 6 to 10 peaks, which may consist of charge state 1 and charge state 2. Generally, I have observed only these 2 charge state for small metabolites, generally charge state 1 and charge state 2. And other ADACs, so different ADACs. So definitely, one peak, one compound corresponding to around 10 peaks. Finally, it's it's kind of it's going to it really tests the I would say elasticity of the data metabolite. There are only around 2,000 to 3,000 metabolites expected, but at your hand you get around 75,000 peaks. A big data which really doesn't provide you any information, but really confuse the user on which peak to be selected and what to be analyzed. And if you go ahead with statistical analysis of that redundant data, definitely you are not going to get any meaningful statistical results, whether be it principal component analysis, whether be it PLSDA, which is again supervised technique for categorizing sample, which is again dangerous if the person is not that experienced as in statistics. This data reduction is important. So we were able to reduce the data by combining all the ADACs and charge stat, and we finally were able to bring down the data size into 3,000 rows. Retention time alignment. There are 2 samples, RANSAC and GAL-Shapley we have implemented. Again, there are many retention time alignment techniques. I have at least found around 10 published techniques for retention time alignment. So again, we thought that these 2 are the most effective. And review picks. Okay, this is manual. This is also another. And this is what I'm talking about, removing contaminants from the sample. So one is the sample and another is the blank. Okay. If you directly try to subtract, say if someone is using Thermos Excalibur software interface to visualize the data, if you try to extract or if you try to subtract this blank from the sample data, and if you want to go for peak detection and peak picking, then definitely it will give you an erroneous sample, because not many, even though there will be many peaks, this blank may not be as clean as it looks like here. So there will be many overlapping peaks, which seems to be observed also in the sample and also in the sample. In that case, Excalibur or any other software, they may not be able to differentiate which one is contaminant and which one is actual pig. So that mess up happens when data is not as clean as like this. So what we did in the SIMMET software is we performed feature identification for both blank as well as the sample, and once features are detected and retention time alignment is done, then we remove those pigs which are aligned to pigs identified in the blank sample. So this is the result we get. We identify alanine and trigonalines. Okay, this trigonalline is quite intense. If we look at the number of counts, we observe it's around 2 million, and another for alanine is around only 88 counts. So SIMMET algorithm was able to detect peak which is very low in terms of its count, as well as very high, that is 4-fold sense. So this shows that our algorithm was capable of identifying what noise is from that of actual peaks. This is the principal component analysis and score plot and loading plot. I have used only one depiction of what we have observed here. 195 is caffeine and 138 is trigonalline. So 195, as we know that, okay, this is caffeine in coffee sample, it becomes non-informative for us to, again, because many research has been done, and many categorizations of the coffee samples based on caffeine has already been there. So what we did was we tried to check, apart from this caffeine, what other metabolites come into play in differentiating this coffee sample. So we remove this 195 from the study. It's very easy in SIMMET. You just deselect the pig which correspond to 195 and then rerun the PCA, and it gives you the whole different perspective of PCA analysis again. So these coffee samples are reclassified on the basis of remaining metabolites, which is void of caffeine, and we found two metabolites. One is 138 still trigonalline, and another 124. Okay, nicotinic acid, that is nicotinic acid. So these metabolites turns out to be the most important metabolites which are capable of differentiating the coffee samples. So metabolite identifications. So let me go a little bit quickly here. So metabolite identification, as we on the basis of exact mass search, we use like 5 ppm to remove isomers. So even then, we have around at least 4 to 5 metabolites for a particular mg value, which really confuse us that, okay, which one is the actual metabolite. So finally we go for MSMS identification. And MSMS identification, as I have just explained, we use NIST standard MSMS database, and we use a proprietary scoring algorithm to identify which scan belong to, which scan is most proximate to the standard scan MSMS spectrum. Then we found out that for this is for caffeine, and I think this is another example. And for nicotinic acid, this is the MS spectra, and we correctly identified that, okay, this is the this is the metabolite which corresponds to the 124 mg, and similarly using this we have identified trigonalline. So I haven't shown the MSMS spectra for trigonalline. And in conclusion, so it's a high throughput software tool which is capable of metabolite identification after thorough peak picking and peak detections, and further statistical analysis is also supported. And this reliable, they definitely it supports the data analysis steps which have not been supported by other softwares. Like there are Progenesis QI from Waters, which is capable of performing statistical analysis very beautifully, but they don't do the database search. You have to go to this one Matlin database to perform that identification. And again, Matlin has only 500 mg values that can be inputted in search. So that's a limitation. And again, XCMS is also a freeware tool, but XCMS XCMS doesn't perform statistical analysis like I think It doesn't support MSMS identification. And analyst, it supports beautiful statistical analysis, but it doesn't support the LCMS data processing. In fact, which is one of the most important steps for successful statistical analysis. So this is the okay, this is the reference papers I have referred to while developing this software. And I acknowledge the contributions of Dr. Aiko Bars from Bruker Daltonics and development team at Biosoft. Thank you very much.
Any questions for Dr. Mayte? Yeah, please. Yeah.
Sure, please.
What are the number of metabolites that it can identify? Like, what is the maximum?
In the metabolome itself, or from
like
the software perspective. Are you
construction
asking
of the
from the software perspective, or
Sorry, say that again.
is that from the perspective of the software or from the technology itself?
No, from the software.
Okay, software, there is no limitations on how many metabolites you can
Like,
identify.
but is there a maximum and a minimum number that you have found? Like, you can say, like, this many number of metabolites we have found. Like,
I have come across many research papers on liquid chromatography mass spectrometry workflows, which says that as compared to NMR, it has the capability to identify around at the most 1,000 metabolites, not more than 1,500 metabolites. So definitely, if I see much more like 10,000 metabolites, I don't think I will expect that kind of number of metabolites from a biological sample.
okay.
So theoretical point of view, we don't restrict that, okay, this number of metabolites can only be identified in one run. But I don't think I will encounter, such numbers of metabolite identification. And in fact, if you look at the NIST database, MSMS database, I think which is one of the most reliable database, it has only around 9,000, compounds in its database.
That's what I wanted to know.
And
How many compounds you have?
yes,
Yeah.
9,000. And out of that 9,000 compounds, 112 compounds are glycans, and around 1,200 or 1,000. I have actually, I'm just I cannot really remember the statistics of lipids. And only few, like around 6,000, compounds are actually endogenous metabolites.
Okay.
So definitely, based on, this one, NIST database, you can identify at most around 6,000 metabolites.
And are those metabolites based on different species? Is there a cross-species
Yes,
reference?
we have a database. We painfully curate the database, like we relate those metabolites to its biological source, kingdom, class, and category.
Okay, so you have a cross-species
Yes,
reference.
you can always filter, source of the metabolite based on, what category it comes from, what kind of biological it was, identified from, or any, biological sample like urine, blood, which are also available in human data, metabolome database, yeast metabolome database, and
Okay, so you have
drug
a
metabolome
cross-reference.
database. So that kind of interlinking between the database has already been done in our software.
And does it identify outliers?
Definitely, statistical, the whole, purpose of performing all statistical analysis like ANOVA, t-test, principal component analysis, these are just to identify what outliers are.
And say, for example, it couldn't find out like what metabolite it is, but there's like the value of the particular metabolite is higher.
Yes.
So in that case, what would be the outcome?
I would leave it to the users because, yeah, there are many response curves, identified, provided by SimMatch software. So from those response curves, you can always visualize, how a particular metabolite is expressed in different biological samples. So even though it is not identified by the algorithm or it's not in the database, so that software has limitation to identify that metabolite, you can always see that okay, the expression level of far higher in this sample, and it may be of interest to you. So you can extract it and. No, that's a proprietary algorithm. I mean, in fact, we have just filed it for patent also, so it's under review.
Okay, please. Okay.
Okay, I have just one comment.
Sure, please.
Since it came that how many compounds or metabolites are there,
Yeah.
we also keep track that how many are there. So
Yeah.
in general, biologically related,
Yeah.
4,000, 5,000 are
Yeah,
there.
that's
But cancer associated only 240 to 260.
Yeah.
This is not in 1,000.
Yeah, that's what I know.
Differentially
Yeah.
expressed in cancer, different kinds of cancer, all solid tumor and other tumor, they are 240 or 250.
Yeah.
So many times we are suggesting that instead of doing targeted
big,
approach,
yeah.
you do all
All
cancer
non-targeted.
associated. And NIST is our place where they make standards for us
Yes.
for spiking.
Yeah, I I get to know, and I was in touch with Steve Steen. Really, I think you are in touch with Steve Steen too.
Yes.
So I have been working with Steve Steen, and in fact, this sample data I have shown to him, and there are many publications which shows that coffee is the biological sample which consists of maximum number of metabolites, which is over 1,000 metabolites in the sample.
But
So-
at the same time,
Yeah.
in these kinds of meetings, if you show some biological sample analysis like serum plasma, that will be better.
Yeah, I
instead
have
of caffeine.
Yeah, sure. I have samples from liver plasma extracts. That is for lipid only. They have done lipid extraction, and I have huge data from those lipid extracts too. And I think we have meat samples, brand samples related to other brands. So those are very very specific to lipidomics workflow. So I wanted to show a general metabolomics workflow wherein we can show that not only lipidomics analysis, which is somewhat limited to lipid extraction. I mean, there are additional techniques for lipid extractions. So I wanted to show that this is a general metabolomics study, and this is what we have done. There are many available software platforms which try to help researchers, but at the same time they are still unable to achieve what people are looking for. Please.
So metabolite identification
Yes.
is a very critical step.
Yes.
And also can be very difficult.
Yes.
So I would like you to comment on in case you are unable to match all the peaks, the fragmented peaks with a compound. Like you have metabolite,
Yeah.
they have many fragmented peaks in your, I would say, mass spectroscopy, right?
Mm-hmm. Yeah.
If some of the peaks are overlapped with other metabolites,
Yes.
and so this is one case, and also in case one peak you cannot find it, how do you deal with those two cases?
Very interesting question. In fact, I mean, this is one of the problem I keep on facing. I mean, running into again and again when we this algorithm. So that's why finally we have turned our face to NIST database to provide a consensus spectra on for those metabolites. So for like caffeine, this particular caffeine NIST has around 120 MSMS spectra, and those MSMS spectra are acquired using different adducts, different instrumentation, different energy level. This one collision energy level, different in source voltage, this one energies, and different setups. And again, for each experimental setup, they have like hundreds and hundreds of spectra, from which they come up to the consensus spectra. So what we did was we have a list of ions, Mg tandem mass spectrometry ions, which are observed in majority of the experimental setups. If a particular, this one is if a particular peak is observed peak is missed out, then we give a penalty. Okay.
Yeah, but in practice, under one level may only have one or two kind of instrument acquiring the data. You have a bigger database that's very helpful, but the experimental spectra you only have those many peaks.
Please repeat the question again.
Yeah.
I think I was.
So experimental spectra. So you have a set of peaks, right?
Yes.
So that's you are going to deal with.
Yes.
Then you based on this experimental spectra, and then you somehow compare with your database, and from your database you decide the metabolite.
Yes.
Yeah.
So there are two algorithms here. One is pattern matching and one is ion matching. So the pattern matching is the algorithm which NIST is following. That means you have an observed spectrum and a standard database spectrum. So they check whether the increase the pattern which you observe in the standard database is same as the pattern which you observe in the acquired spectrum. So generally, I think most probably, I don't know what algorithm they use, but most probably they are using some Spearman's rank correlation based algorithm mechanism, because when I try to recalculate the score they provide, I think that's very close to what I observed. So definitely they are using some correlation based algorithm technique, which says that a particular pattern is observed, then they bring that metabolite up and they said that, okay, this is the metabolite. But that's not the case in actual life. Even though you acquire, you use the same instrumentation setting, the pattern may be different. And even say a researcher restrict his mass range from, for caffeine, I restrict the mass range from somewhere to 110 to 200. And I observe only 3 or 4 peaks in my mass range. But in the database, there are many instrumentations setting which are same as mine, but the mass range is different. So their mass range starts from somewhere around mg value of 40 and it goes up to 200. So there are even some dynastic ions observed at the very lower mg values of 40s and 53 and then 110 and then 124 and then 138 and the final one is at around 194. So the pattern is never going to be a sufficient one. That's why we come up with our scoring, proprietary scoring algorithm, which says that, okay, these are the probable ions and the evidence of this particular ion as a dynastic ion for this particular compound has been verified from this spectra. And we have populated those ions from hundreds and hundreds of spectra, and then we say that if a particular dynastic ion is missed out from your observed spectrum, irrespective of what instrumentation setting you set, we give a penalty. So definitely I can, in a very simple language, I can summarize that our algorithm is a penalty-based algorithm, which gives some sort of weightage when there is a dynastic ion, and which penalty when there is miss out of any dynastic ion. So it's not perfect, I can understand. There is always case of false positives despite our best effort, and our effort is to improve that algorithm, algorithm in future.
Okay, we have the last question. Yeah, okay, please.
Thank you very much for your presentation. It was very, very interesting. I saw that you have some collaboration with Bulker, for example. Bulker. And do you have some collaboration with other
yes,
constructors, such as Thermo Fisher?
Yeah.
And is it possible to also process raw data from Thermo, for example?
Yes.
Okay.
You can, I have, we have already distributed the DVD, wherein you can install the SimMath software and SimLipid software. So we have different icons. Say you can import raw data file from Thermo, WIP file from AVSIX, T2D file from AVSIX, this one, MALDI, MSMS workflows. Brokers data like BAB, YEF, FIT, and even we are in the process of supporting SimMath data file that is .lcd.
Okay, and is it possible also to plug our own database to enrich, to have a kind of custom database?
Yes. Our facility is available. That enterprise edition server is available. It can be installed at client's end, and if you want to append, or if you want to add additional spectra or additional metabolites into the database, and you can organize your database as per your wish, then you can do it using our software.
Okay, thank you very much.
Okay, thank you very much.
Thank you. I'm sure we can always have a very close discussion when Dr. Hiko is available for the tea. Okay, thank you. Now we'll go on to next speaker, who is Dr. Lee. Comes from the well-known, the national or wonderful institution in Asia called National University of Singapore. Moreover, is well known in the research of what you call metabolomics, where I'm sure the topic, I'm sure he'll deliver a wonderful lecture on application of metabolomics for toxicity assessment of solid waste reutilization. It looks as if he'll try to enrich or he'll try to combine more than one domain, like toxicogenomics or toxicometabolomics and biotechnology and bioinformatics, etc., etc. I'm sure. Please, sir. I'm sure it will be nice if you could enrich our knowledge on these, wonderful domains. Okay, finally. Yes, sir. Thank you, sir.
Before Dr. Lee makes his presentation, quick announcement. Any presenter who has not received their certificate can go out to the table right in the hallway and pick it up anytime before noon today.
Thank you, sir. Yes, sir. Dr.
Hi.
Lee.
Good morning. Yeah. So please collect your certificate later on if you haven't done that yet. Anyway, for this morning, I just share with you some results from our work with our collaborators in the Faculty of Engineering, working on some waste reutilization. So the topic is actually analysis of leachate from gasification of solid waste by metabolomics techniques. My students, Sini and Wan Ning, are working on this topic, and then our collaborators, Dr. Wong, Menrung, and Professor Wang, and Professor Nia from Faculty of Engineering. Normally, when we have waste, we either put it in the landfill and then forget about it. Then, but that's the limitation of the land available for doing that. So a lot of times you do incineration. When you do incineration, you burn everything completely. Whereas in gasification, what we try to do is we don't really burn all the materials. We gasify it so that some of it can still be reused as a fuel. So in that sense, we are trying to reuse the waste to recover some of the energy. However, in this process, you get some waste products as well. The fly ash, bottom ash. The fly ash is the more lighter component. The bottom ash is the heavier one. Then you have the boiler slag because you are still supplying heat, and then the flue gas desulfurization material to remove the sulfur. Still disposed in landfill, so we try to minimize the generation of the waste, the fly ash, the bottom ash, by reusing and recycling them. One of the common ways using this material is construction materials, cement, concrete, and so on, or some just used as soil stabilization, mine reclamation, and so on. However, when you put this back as construction material, then there's, when it rains, when it gets wet, then some toxic materials can leach out. So it can be a potential pollution problem. We try to reduce the impact of this leaching effect and the cost of treating this kind of pollution problems. So metabolomics, in fact, this is from trash. You try to make some ash, and then after that you get some cash out of it. The composition of this ash materials are actually quite complicated. Yes, some of it is inorganic, so you have all this silicon dioxide, oxides, hydroxides, and so on, many elements. And then there's also organic contents. Some even have dioxins and pH, which are actually from the incomplete combustion, because the gasification is not complete incineration. Then there are many chemical analysis they can do to analyze all the things, the polycyclic aromatic hydrocarbons, also the dissolved organic carbons, and so on. However, when you just do chemical analysis, you may not really fully understand the toxicological effects. So in this study, we try to combine both aspects, doing both the chemical analysis as well as to analyze how these toxic compounds might affect living organisms. So this is the ecotoxicity study that we did. We collected the bottom ash and fly ash. First, we do the chemical characterization of the leachate that we leach out from the materials. These are the ash material, using a variety of analytical techniques. After doing the chemical characterization, we compare with some guidelines from EPA and all this agency to see whether they exceed certain levels. When they are not, then they are okay. Whereas for the ecotoxicity, toxicological study, we used this small water bugs. They're called Daphnia, and then we see how it affects these organisms. If it's above certain levels, then this is called ecotoxic material. When they're not, then they probably would not be so toxic. This aspect currently is not so well established. So in fact, what we are trying to do is also to establish the kind of potential guideline that can be used as a way to do these toxicological studies. The samples, sort of from different sources. One is from a local company. Sample A is a bottom ash. B is a fly ash. C is some of the sample that we made ourselves, bottom ash. D also, they're slightly different, but kind of very similar. That actually allow us to do some comparison. And then sample E is from the Public Utilities Board because they do water treatment, and then they also collect some of this sewage waste that needs to be treated. So we try to see together what the effects will be like on the leaching. This is the real workflow, which is firstly the preparation of the leachate. Then we do the elemental analysis. For the mass spec analysis, we do first some extraction. We do both GCMS and LCMS, just screen for both volatile and non-volatile components. For the toxicological studies, you use this water fleas, and then the metabolomics allow us to really characterize all the different toxicological effects on these small organisms. Sini did most of the mass spec analysis, and Wenning did most of these biological studies. So from the elemental analysis, you can actually get profiles of all the metal elements, some of the major elements, some of the trace elements. Then you can see which ones are present at higher concentration in which of the samples. So we find that in these two fly ash potassium for some reason. So we're looking for those. And also we can actually see whether some of these trace elements are present or not. ND is just not detecting that below the detectable concentrations. Then we do the untargeted analysis using LC, quadruple time of flight mass spectrometry. These are the positive mode and negative mode ionization conditions. These are the HPLC conditions. For the positive mode, for the different ash samples, these are the typical chromatograms that you get. And then for the negative mode, then you get this typical chromatograms. Then you can do your metabolomics, PCA analysis, and so on. The sewage sample is very different from the ash sample, so it's well separated. The blank, they are not containing this waste material. Again, they're well separated. And then the bottom ash here, A, C, D are the bottom ash, and then B is the fly ash. So you can see they're kind of reasonably well separated. The sludge actually especially is well separated from the ash. And then when we take out the sludge sample, just looking at the ash samples, then the bottom ash all clustering together, and then separate out quite well from the bottom ash. So in that sense, you can really have a segregation of fly ash from the bottom ash leachate. That was in the positive ionization mode. This is the negative mode. Again, the sewage sludge is well separated from the ash sample. A, C, and D are the bottom ash, and B is the fly ash. Again, you can see very good separation of the sludge from the ash leachate, and it's quite possible to differentiate different sources and nature of the leachate that giving all this data. And just look at the ash samples. So these are the bottom ash samples, and this is fly ash samples. And again, you can see very good separation of the fly ash from the bottom ash leachate. And also, you can really use this result to differentiate the source and nature of the leachate. After that, you can actually, find out what are the toxic components, and you just make tables of all these different. So this is a very big table, so I won't go into details, but we can actually group them into all different types of toxic materials. So next we move on to the toxicological assay using this aquatic organism. Basically, we want to study the effect of the leachate on this Daphnia. There are some common methods used. The use of algae. But in our case, we use this decreased mobility of this water flea. These are about a couple of millimeters size. See it visually, so when it's not swimming very well, then you can tell they are not feeling well. Okay, so that's the approach that we're taking. This actually is a well-used test organism for acute and chronic chemical test. For acute testing, basically we look at the mobility within the 48 hours period. In the case of chronic test, it's the reproduction mechanism that is affected over a 21 period. So in that sense, there's a procedure that you can follow to do this test. The reason we use this Daphnia is because they're quite easy to culture. The students are actually growing tanks of these small fleas. And then they eat on microalgae, which is being used for some of our other experiments as well. And the life cycle is relatively short, and they can grow in high densities. So what we did is that we followed the ISO procedure for acute leachate toxicity testing for this Daphnia samples. For each concentration, we use just 5 Daphnia, and then the 6 concentration being tested here. And you have to keep them in the standard conditions. So these are the standard conditions that we test, and then we measure all the parameters that are of significance. Why it's not moving? It should. Well, anyway, I think the computer is not really making them move. Basically, this one should be still swimming very well, okay? And this one should be, just move a bit and then sleep for a little bit. But anyway, so this after they are exposed to the leachate, they would not be moving very well. Okay, it's like the kids when they're young and healthy, they're running around. When they're not well, then they just lie around somewhere. Okay, basically that's what you should be seeing. Anyway, then we actually count which sample is causing how many of this Daphnia not moving so well, and then you can total up the data. So there are 48-hour concentrations. There's no immobilization at the control and this very dilute solutions. In the case of solution, 6 out of the that was sick, and then 16 were immobilized at the 5-fold condition. So when the literature in the conditions, so we just do this PCA analysis again. So the immobilized definitely clearly have different also use this to really understand how the metabolism is affected. So the students are currently working on the detailed analysis, but the thing is that our collaborators, they're more interested in the testing procedure. So we can kind of tell them that the method can work. This immobilization test can be the toxicity effect of this. So in conclusion, this non-targeted screening by LCMS give you valuable toxicological information on the literature samples. You can do full characterization of the toxic components present. The method can be used to determine which organic compounds can contribute to the toxicity of the leachate, because you have all the data from your mass spec analysis. The Daphnia magna, this water-free, can be a useful alternative method to monitor water quality, and as a quick screening method to test the toxicity of some of these waste that you try to reutilize. Okay, yeah, thank you. Yeah, thanks.
Now the session is open for the questions.
Yes,
questions from the audience.
Okay.
Thank you very much for your very interesting talk.
Okay,
as regard for the toxicity and for those particulates, because for the waste they have maybe have nanoparticles. Those might be very toxic, cause lots of toxicity to the your test for the aquatic species test, right? And also, you have a great separation for the immobilized species with sample A to D. So this, you mentioned that they are the metabolites. They are significantly different. What are those metabolites? They are different.
Okay. Firstly, about the nanoparticles. For this study, we actually did not really try to detect the nanoparticles. For that, we had to use another method. We use single particle ICP-MS to do that. But for this study, we just want to see the overall toxicity effect. Whereas which metabolites are different in the Daphnia, we are still working on that part. So this is the stage we are at now. Okay. We analyze all the leachate already, but at the moment we know the difference, but we haven't gone into details yet. Okay. Okay. Oops. I think it's this one. Yeah.
Okay. My question is about the organism.
Yeah.
That you are using, or that using to test the toxicity. I mean, the people working on toxicity have standard organisms like Drosophila, mice, rats, and all these kind of tests, they have detailed data. So I wonder why you are using this kind of organism.
Okay. Well, there are many different tests that you can do, but I think in terms of the reusing of these, they wanted to have something that is quick enough. So 48 hours is the range that they're happy with. So for other things that take longer, for example, you can do. Some people use bacteria, then you have to do the culturing and all that, and also the effect may not be as clear. There are other tests that we are doing using the flies as well, but it seems that the Daphnia give us a better indication. So yes, you have a choice of other method. In fact, for reutilization, they don't really have official guideline what tests you need to do. So we are trying to establish a kind of guideline also. Yeah. Yeah.
Easy method, easy. So we
something
easy
quick and easy and reliable.
Method.
Yeah.
That's why we use what we call conceptual model organisms.
That's right. Yeah. Okay. Yeah. Yeah.
Little foreign.
Yeah, that's right. That's right. Mm-hmm. Yeah. Yeah. Right. That's right. Yeah. Mm-hmm. Right. Yeah. Right. 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 Yeah. Actually, what we use is also an approved method. It's called ISO, the European organization. Right. Right. Yeah. That's right. Okay.
Okay. Very interesting. Thank you,
Yes,
Dr.
okay.
Lee. And also, I think it has great significance.
Mm.
I think my question is a little bit similar with your question. Is
Yeah.
I'm curious about the model you use, the Definier.
Mm.
So, to my understanding, if I don't know if the Definier is a well-established model for the assessment of the toxicity analysis, and if there is some kind of situation that this kind of flea can develop a resistance to some kind of the toxicity, like the bacteria has a resistance to
Okay.
the
Right.
antibiotics.
Okay.
Yeah, thank you.
Okay. What we are looking at here is not human toxicity. So, so this is a slightly different focus that we have. Yeah. So eventually, okay. All right. Yes. So anyway, sorry, I forgot what your main question is.
My first question is if the Definier model is well established,
Yes.
and the second is about if there the existence of the maybe the resistance of the Definier to certain time of the toxicity
Yeah,
in the
okay.
West. Yeah.
Okay, I know bacteria resistance is more well known, but for organisms, I think they haven't really looked into that yet. At the moment, I think people haven't really studied how that can become resistant to exposure. No, I don't think so. Okay.
Same direction.
Yeah.
Did you monitor sulfur compound, your compounds?
Sorry, sir.
Sulfur.
Sorry.
I think we didn't really focus on it, but it's all in the data.
Yeah,
Okay. But we
because-
actually haven't really focused on it because it hasn't really stood out as the major metabolite.
Because your ecotox test is extremely sensitive to these compounds and could over results for the ecotox.
Right. Yes. Okay. Okay. Right.
That's all. If no other question, I'm sure we'll thank Dr. Lee for enriching our knowledge on eco-metabolomics. That's what wonderful talk this time. And then it's time for us to the chat. Okay, shall we go? Okay. Thank
Do we have a tea break? Do we have a tea break? Do we have a tea break? No tea break. No, after that. Maybe 5 minutes. Yes, sir. Yes, a tea break. We'll honor Dr. Lee. Okay. Sorry. Tea break. Sorry. Just excuse me. We'll try to invite all the speakers who are given this month. Few minutes. Okay. All right. Okay. Thank you. Thank you. Okay. Thank you. Thank you. Okay. Thank you. Honor me. Okay. Good. Thank you. Okay. Thank you. And speakers will. Okay. Dr. Lafigran. Sorry. It just went out. Okay. Okay. Yeah. And Sanjeev Metari. Maybe in the next session. Maybe when they come back. No, 3 or 4 this one, right? Okay, next. Okay. Okay. Will be honored. Yeah. Thank you. Thank you. Okay. We'll break for the tea now. We'll try to meet after 15 minutes. Yeah, 5 minutes, I'm sure. Thank you. Thank you. Okay. There'll be 3 talks. All right, okay, no problem with me. Okay, so there's a change in the order of the presentation. So the first talk will be given by the third speaker on the original program. And the speaker is Mr. Narinder Singh, yeah, okay, from Deakin University in Australia. It's a long way from here. Okay, you want to introduce your topic? Yeah, okay, please.
Good morning to all of you. Today I'm here to present a very interesting topic on glioblastoma called dedifferentiation. Probably it's not directly linked to the metabolomics about this conference, but some of this aspect which we are going to do in future will be having a direct link with this metabolomics. So I'm going to talk about dedifferentiation. It's a very critical phenomenon in glioblastoma, which helps in movement of its grade from lower grade to higher grade of the tumor. So I start with the introduction. Firstly, glioblastoma is a sort of brain tumor. It's a very highly malignant brain tumor. So, and the mortality rate is quite low. So most of the people diagnosed with the high grade gliomas have got a lifespan of not more up 1.5 years to 2 years. As mentioned about the different grades. So grade 4 is the, there are different sort of grades. So grade 4 is called the glioblastoma, that is also called the grade 4 astrocytomas, because the astrocytes are the type of cells in the brain. So the tumor of those cells are called astrocytes, and the grade 4 astrocytes is considered as a grade glioblastoma. So different grades as I mentioned. So it's I'm talking about the Australian concert. It costs like nearly $150,000 per person. This is the most amount of money they are spending on treating this disease, according to the Cancer Council New South Wales in Australia. And also this is the highest cause of cancer death in person aged less than 40. So this is a tumor basically present in the younger people. Also, although this tumor also present in the higher in the older ages, can tumor. And then why this glioblastoma is so malignant? All the solid tissues are very malignant, but considering the site of disease called brain is being a very sensitive organ, so it's very hard to treat this tumor, and because of the malignancy, the higher malignancy of this tumor. The thing is, it's got a very high ability to relapse, and because of the resistance to the anti-growth molecules like drug and proteins, and they have got a high efficiency to avoid apoptosis, and also ability to self-transform into more malignant phenotype. So this is the actually topic. This is the point I want to discuss in detail during my presentation. And also it has got very high efficiency of high potential of invading into tissues and metastatic potential due to the hypoxia and genetic aberration. So hypoxia, it's a very complex term. It's a very broad term. So which to the different complex phenotypes in the tumor called angiogenesis and all, it's a very complex. So this hypoxia actually helps in the introduction of or the ability to transform the tumor from the lower grade to higher grade. So that is what we have actually observed in our studies. And so there is a different upregulation, different growth factors like EGFR, connective tissue growth factors, PDGFR, etc., and VEGF I have already discussed, and the upregulation of various cancer stem cell markers like CD133, CD44, SOX2. SOX2 is another pluripotent stem cell marker. This is considered as a differentiating marker. So this, even though this CD133 and CD44 and SOX2 are the broader cancer stem cell markers, however, SOX2, ABCG2, OLIG2, and A2B5 is also called CD15. So these are some specific cancer stem cell marker to the brain. So coming to dedifferentiation process in glioblastoma, so this is a very critical aspect of differentiated tumors. So they got a very high capacity to dedifferentiate from lower grade to higher grade. So the process of the meaning of dedifferentiation has actually is the conversion of the actual differentiated tumor cells to the more stem cell sort of phenotype. So that stem cell phenotype, according to the stimuli provided or stimuli given in the environment, can actually differentiate to either to neurons, astrocytes, or either to the more complex phenotype. So I'll come to the point in some time. So as I discussed, the tumor differentiation depends on various factors like hypoxia, genetic variations, and grade of the tumor. MicroRNAs are the other elements which can help in the induction of dedifferentiation process in glioblastoma. So there is no current therapy available to treat the actual dedifferentiation signaling pathways in glioblastoma, although there are different inhibitors or different anti-cancer drugs which can treat the basic aspect of the glioblastoma. And also and another aspect of dedifferentiation is that conversion of dedifferentiated cells into more angiogenic sort of cells when they are growing under more hypoxic condition and more nutritionally stressed conditions, because of the when you grow the cells in the more hypoxic condition, like 1% and 1% rich conditions. So that actually we have observed in our cells that these cells actually take the form of pericytes, which actually support the blood vessels. These are elongated cells, so around the blood vessels in the tumor and all across the body. And so we have also seen the expression of HIF1. HIF1 is a protein which in normoxic conditions is ubiquitinated to the and it degraded. And but in these cells we have observed the HIF1 expression even in the normoxic conditions. So present study. So this is the first in vitro study that I am aware of, and we are aware of, that in our culture normal condition, 2D culture condition, this is our cells. The lower field is the cells like differentiated CCF STTG cells, which is this is the present cells, like we were working on these cells. But actually these cells like dedifferentiate self into the upper morphology. So there is a significant difference in the morphology of these two cells. As I've mentioned, that dedifferentiated cells resemble more sort of stem cells. So and thus they express very high expression of various cancer stem cell markers. So this is my study. So nobody has actually observed, like this is serendipity. So we are working with the cell lines, this present cell line, and then suddenly this cell line dedifferentiate. So that is the only in vitro study across the world that has been reported out. And then to confirm the astrocytic origin of these cells, because the parent cell line was the astrocytic, so we want to see whether the dedifferentiated cells which have evolved from cells are actually from the astrocytes, are from the parental origin. To confirm the expression, we have seen the expression of GFAP and S100 beta, which are exclusively astrocytic markers. So the expression of both the proteins are confirmed with the help of immunofluorescence in the top figure and with the help of Western blot. And another interesting findings we have seen is that the expression of GFAP was significantly upregulated in the case of dedifferentiated cells. That this is the literature shows that the expression of GFAP increased when there is any insult in the astrocytes. So when astrocytes become more reactive, so they actually generate high expression of GFAP. So we consider that due to some stress or due to reactivity of astrocytes, these cell lines differentiate actually into dedifferentiated cells. And then we check the expression of various cancer stem cell marker in both the cell lines. You can see, and this is like ABCG2. ABCG2 is a class of protein, of PGP class of protein, which helps in the efflux of any drug from the cell into the tumor microenvironment. So this actually protein is responsible for the drug resistance in lot of tumors, including the glioblastoma. So you can see that in comparison to the dedifferentiated cells, the expression of ABCG2 is quite high, and the same for the CD133. CD133 is highly regarded as a cancer stem cell marker universally. So and also glioblastoma is a markedly high expression of CD133, ABCG2, and integrin alpha, alpha 6 is a in the migration of the tumor from one side to the secondary side. So you can see that the expression of quite as compared to the differentiated cells. You can see that in the case of this differentiated cells, we can see that the globular morphology that shows that even in the differentiated cell we can have differentiated cell, because that this cell might be a cancer stem cell marker even in the case of normal differentiated CCF cells. And then the survivin is regarded as a highly potent inhibitor of apoptosis protein in cancer, universally regarded as a protein. So the expression of survivin is quite high, even though the expression is like similar in both expression, because this is exclusively cancer-specific protein. So we do not find any expression of survivin in the normal cells apart from the embryonic cells and actively dividing endometrium cells. So there is no expression of survivin was observed to date in the normal cells. So this survivin is highly regarded as a cancer-specific marker. And then we check the migratory potential of these cells through transwells and through ECM. As you can see, that lower panel corresponds to the migration of dedifferentiated cells through transwell in 24 hours. You can see that these are the elongated cells, these are the differentiated cells, and this is the dedifferentiated cells. You can see that a very high migratory potential was observed in the case of dedifferentiated cells, and the same for the invasion. Invasion is through ECM. ECM is the extracellular matrix proteins, consist of various collagen, glycogen, all proteins, which helps in the. So this is actually matrix. So what tumor cells do is tumor cell actually express various MMP proteins, metallic metalloproteases, which helps to degrade this ECM. So we can assume that the expression of this MMPs is quite high in case of this dedifferentiated cells as compared to the normal differentiated cells. And then this is the a very conductive experiment to confirm the angiogenic potential of the cells. So what I did is, so actually I designed this experiment just from the literature. So what I did is, I just put a like ECM mixture in the 96-well plates, and then count the number of cells, and just put the like 30,000 cells, 20,000 cells, and 10,000 cells. So what I have observed is that tubule formation is a very critical phenomena in the case of any tumor. So you can see that within 24 hours, when I incubated 30,000 dedifferentiated cells, there is a formation of network-like structure, as you can see from here. There is a high tubule forming capacity was observed in the case of dedifferentiated, but not in the case of differentiated cells. As I have reduced the number of cells, so the tubule formation capacity was significantly reduced in the case of both the cells, as I have not observed any tubular formation in the case of differentiated cells. So this is the photograph of the human microvascular endothelial cells that is taken as a reference image in this case, just to show that these cells sport behave like endothelial cells in the tumor. And then we carried out various hypoxic studies to check the angiogenic potential of these cells. So we checked the baseline expression of VEGF. VEGF is an angiogenic marker that's regarded as a universal angiogenic marker. So what I did is, this is the first panel is of 1% O2 conditions. So normally we grow the cells in the 5% O2 conditions, but this is the 1% hypoxic conditions, 1% O2 conditions that resembles the hypoxic condition in the body. So what I observed is that when we grow the cells in the 1% hypoxic condition and the 10% FV condition, like proper medium condition, but in hypoxic conditions, I have seen that a change in the morphology of dedifferentiated cells, but not in the case of differentiated cells in the 1% hypoxic condition. And also even though the expression is pretty much in the case of in the more nucleus of VG, the 1% and the lower cells were growing hypoxic stress, and it's how the cells work in the how behave in the very nutritional and physical deprived conditions. So we can see that in the case of 1% 1% FBS conditions in the lower panel, you can see that the more tubular, as I've observed in the last slide, more sort of tubular network structure was observed, and the expression of VEGF also got significantly increased, and more VEGF expression was, more nuclear expression of VEGF was observed when grow in the nuclear. That means that VEGF tend to localize in the nucleus, that that may have implication with the hypoxia, because VEGF in a hypoxic regulates promoter region, so that helps in the driven of these cells into more sort of angiogenic path behavior. And then what we did is alpha SMA. We checked the expression of alpha SMA. Alpha SMA is a marker for pericytes. Pericytes are the cells which surround the blood vessel across the in the body. So we have checked the expression of alpha SMA with the help of immunocytochemistry. So we can see that the baseline expression of alpha SMA in the differentiated cells when they grown in the 1% hypoxic condition and 10% FBS condition. So there's not much expression of alpha SMA, but however, the expression of alpha SMA in the dedifferentiated cells is very high. So not every cell is expressing alpha SMA in the top right hand panel of CCF dedifferentiated cells. That shows that the cells those are expressing alpha SMA has got more tendency to support angiogenic vessels in the case of tumor. And then when you introduce the 1% hypoxic condition and 1% FBS condition, we can see that there is a formation of a clear formation of tubule in the case of even in the differentiated cells, and you can see that the significant change in the morphology of dedifferentiated cells and very high expression of alpha SMA that shows that these cells has got very high angiogenic potential. And then we check the expression of various human phosphokinase array protein with the help of protein array antibody, and then we have checked the 43 different proteins in the same membrane, and then I have plotted here some very important proteins here. So this all P33 alpha is a mitogen kinase protein which is upregulated in the case of which is a protein which is upregulated in the case of stress. So we can see that and this P38 alpha leads to ERK1, which is a downstream regulator of this MAP kinase protein, and the expression of ERK1 and 2 phosphorylated ERK1 and 2 has got a very significantly high expression in case of dedifferentiated cells. Although phosphorylated form of this P53 is a tumor, so for this was very high. We have observed a very high expression of phosphorylated P53. Shows that this is actually a tumor suppressor protein. But when P53 is mutated, this actually leads to gain of function. So gain of function actually helps in the actually not the suppressor, but it then act as a proto-oncogene when there is any mutation in the case of P53. So we are assuming that there is something gone wrong in the P53 gene that has converted these cells into dedifferentiated cells, and they are behaving as highly malignant cells. To confirm this phenomena, we have done the NGS sequencing, next gen sequence DNA sequencing, and we are analyzing the data. So the future results will come. And then certainly another important protein is C-Jun. C-Jun is a protein which helps in the, this is a protein which helps in the jumping genes actually. So jumping genes are, there might be a possibility that there is a very high expression of C-Jun was observed in case of dedifferentiated cells. And this is what we have observed in the case of STR profile. So we have also done the STR profiling of these cells. So there's the involvement of jumping genes, according to the data we have analyzed from the STR. So there is a jumping genes are involved in the case. So this is what we have seen here with the help of this phosphokinase array. The C-Jun plays a major role in these dedifferentiated cells, and also at the same time the expression of HSP63, which is a heat shock protein, also elevated in stress condition, various stress condition in the case of tumor. The expression of HSP is also very high. And then we analyzed the spheroid capability of these cells. Like this is a 3D tumor spheroid, actual in vitro 3D tumor spheroid formation. So this spheroid actually resembles the actual in vivo tumor in case of laboratory experiments. So we have seen that we have inoculated the equal number of cells, 1,000 cells, to form and let the cells to form agarose on agarose coated plates. So we have seen that within day 2, the spheroids by dedifferentiated cells, the spheroids by dedifferentiated, the lower panel is got very compact within day 2. However, in day 2, even though that spheroid is evenly balanced and formed, but the spheroid in the case of dedifferentiated cells is very compact, and we have observed the necrotic zone within the within day 1, within day 2 of this dedifferentiated tumor. So this is actually the case of in vivo conditions. As the tumor progresses from lower grade to higher grade and from lower malignancy to higher malignancy, this necrosis is a major critical phenotype of the tumor. In the high development of necrosis within day 4 of this tumor, and you can see that within day 7 this tumor actually forms budding and helps in the spreading. So that is what we have observed at high migratory and high invasive potential of these cells. And then this is all the hypoxic studies we have done. I have already done that. We have checked the expression of various cancer stem cell markers. You can see that the expression of HIF1 even. So these are the different. This legend shows that when from 1 to 4 is dedifferentiated cells when they grow in a 10% hypoxic condition and 1% hypoxic, 10% FBS conditions, and then 1% FBS and 1% oxygen. The same condition that we have seen for the angiogenesis and SM expression. So we have seen that the baseline expression of various important molecules which are involved in the pathogenesis of glioblastoma. So as you can see that very in the panel third, we can see that very high expression of survivin, which is a critical molecule, and there are literature shows that there is a recent paper published in the Nature that shows that survivin is actually a protein which helps in the progression of tumor from differentiated to more dedifferentiated phenotype and helps and hence more malignant and more drug resistant tumor. And then to see whether these the changes in these cells is actually epigenetic translations or DNA methylation. So what we did is we want to see how, because we have seen the expression of various neural stem cell markers like nestin and other neurofilaments. So we want to see whether these cells actually have a potential of neural stem cell and potential of reverting them back into the various neuronal bodies and astrocytes. So what we did is we have treated these cells with the histone deacetylase inhibitors, which these inhibit the activity of histone deacetylases in the nucleus. So we can see that when we treat these cells with the help of this LBS 589 and SAHA, this is like type 1 and type 4 histone deacetylase inhibitor. We have actually seen the formation of this is actually the structure of neurons. So and when we treat when we treat these cells with the proliferation of these cells is completely stopped, and they are reverting them back into more astrocytic and neuronal cells. So the expression of these neuronal bodies and astrocytes was to confirm whether they are neurons and astrocytes. We have checked the expression of various neuronal and astrocytic markers with the help of immunofluorescence. And then PGCCs. PGCCs is a polychain cancer stem cell marker. This is a very hot topic in cancer. These PGCCs are actually cells which helps in the proliferation of cancer, which helps in the release of that very high expression of the PGCCs are very giant cells, so they have got a bigger size as compared to the normal cells. You can see that very high number of PGCC cells was observed in the case of dedifferentiated cells, and also not in the case of differentiated cells. And this is the right panel is the neurosphere formation. So neurosphere formation is another important attribute of the tumors of the central nervous system. So the high capacity of neural was observed in the serum-free medium conditions. So that shows that these cells are more of stem cell characteristics. So in conclusion, I would like to say that we have observed the first in vitro formation in the laboratory-based cellular-based study. We have observed the transformation of like a cell line to a more advanced and more malign targeted de-differentiation. We confirmed with the various NGS data. Thank you very much. And I would like to thank you about my supervisor, Dr. Rupinder, Professor Jagat, and Professor Colin, along with Professor David Ashley, and AISRF, Australia India Strategic Research Funds, and NHMRC grants. And also, I would thank the organizing committee exhibition on metabolomics.
So you did in vitro work. So whether your de-differentiated cell, where tumorigenesis in mind.
That is what we are doing because I'm in my final year, so we don't have enough time. So we are proceeding to that step actually to check see the tumorigenesis cells in the mice.
Okay. Second question is you get a two cell line, right? And one is differentiated, one is de-differentiated. How you get a differentiated?
So that is what the trigger we don't know now. So these cells, as explained, these cells we observed serendipity. So
this originated from same cell line, right, or different cell line?
So this is origin. These cells originated from the same cell lines. So
if from a same cell line, how you get a de-differentiated cell line?
So this cell line actually evolved itself from the parental differentiated cells. So we have called this as a de-differentiated cells because they are expressing more sort of differentiating markers like SOX2.
Yeah. My question is how you did.
Itself evolved. So there is no any contamination the cell line. So we got this cell line from ATCC. So to rule out all that possibilities.
But you keep before you they automatically de-differentiation, right? How you can keep in different cell?
So they are in a de-differentiated state, growing like a de-differentiated state now.
Okay.
Yeah, they are differentiated state. Okay. So that is what we want to confirm with we have done the next-gen sequencing to actually evolve what are these cells. So to actually evolve the exact genetic makeup of these cells.
Okay, good.
Do you have any idea about if there are differences in production reactive oxygen species, because they are known to be important for hypoxic response and also to activate some of the proteins that you are seeing activated? So I'm just curious.
We have actually not looked at the expression of ROS, reactive oxygen species in the cells, but that might be a future. Once we check the genetic makeup of these cells and see the tumorigenic capacity of these cells, then we'll see, we'll further.
Okay. So the next talk will be given by Maclay. Is that how it's Martinez Weyers from Northwestern University in the USA. You want to introduce the topic?
Yeah. So thank you so much for the invitation and the opportunity to talk about my project today. My name is Sima. I work in Dr. Navdeep Chandel lab at Northwestern University in Chicago. And our main topic of research is the mitochondria. We studied its role in many processes and diseases, such as cancer, stem cells, differentiation, and also immunology. So we thought that it was going to be interesting to see which are actually like the functions of the distinct pathways that the mitochondria is involved. One of the reasons is because we know that mitochondria is involved in tumorigenesis. It's not only the main producer of ATP in the cell, but also is involved in biosynthetic processes. Many of the intermediates of the TCA cycles are precursors for the synthesis of macromolecules. It is also involved in signaling. I will talk about this in a few seconds. And also more recently, the 1-carbon metabolism pathway has been described to be important in tumorigenesis, not only for the synthesis of nucleotides, but also to balance the cells die by. It's very interesting to see how the mitochondria has evolved all these ways to communicate fitness to the rest of the cell. Some of the ways are, for example, we know that when the mitochondria don't produce ATP, AMPK is going to be activated, and that is going to reprogram the whole metabolism of the cell. Also, some of the metabolites in the TCA cycles are known to be able to travel to the nucleus, and they can modify the activity of some enzymes important or responsible for the post-translational modifications of histones. And also reactive oxygen species generated in the complex 3 can go to the cytosol and also activate some of the transcription factors. So all these changes at the end are going to decide what genes are expressed or not. So it has a huge impact in the cellular physiology. But the question we want to answer in this project is what functions of this organelle are necessary for cell proliferation and epigenetics, which are two key events in cancer. We focus in two different activities of this organelle. One is the TCA cycle, which is important for the production of metabolites that are going to participate in the biosynthesis and in the signaling. And then also the membrane potential, which is important for the production of ATP, the production of reactive oxygen species, and also to keep the iron sulfur clusters. So first we want to test if mitochondria is important for these processes, right? And how are we going to do that? We have a very nice system. We are working with 293T cells where we can induce the expression of a dominant negative version of POLG, which is the enzymes involved in the replication of the mitochondrial DNA in a doxycycline-dependent manner. So when this mutant version is expressed, you lose the mitochondrial DNA replication. As the cells divide, they are going to be losing more and more this mitochondrial DNA, and you end up with what we call a row 0 cell with no mitochondrial DNA. We verified that this ectopic mutant version is being expressed in the cells when we treat the cells with doxycycline. We normally do the experiments treating the cell for 3, 6, and 9 days. We confirm by RNA-seq that these cells, when you treat them with the mutant cells, when you treat them with doxycycline, you start losing the mitochondrial transcripts. So you see how by day 6 is like the transcript in mitochondria are very decreased, and by day 9 they are basically gone. Whereas you can see how wild type cells, even treated with doxycycline, the abundance of the mitochondrial transcripts are not affected. So doxycycline by itself is not having an effect. And we also confirm this data at the level of the proteins. As you can see here, if we treat the mutant cells for 6 and 9 days with doxycycline, there is no expression of this mitochondrial encoded protein, COX-2, whereas the expression of this nuclear encoded protein, SADH, is not affected. So the system is working. And very importantly, in the wild type, any of these effects are happening. And then we measured the viability of the cells. I think this is a very key experiment because all the effect that we are going to see after this, we know that they are because they are losing the cells are losing mitochondrial DNA and not because the cells are dying. So it's important for the interpretation that wild type and mutant cells treated with doxycycline and losing mitochondrial functions are totally viable. So when you lose your mitochondrial DNA, you are going to lose the spread of complexes 1, 3, 4, and 5. So the electron transport change is cells that is gradual, and the fact that it's gradual is going to be very interesting to see the mechanisms and the changes that are happening while you are losing mitochondrial function. Also, complexes are not clean, so we also observe that we are losing the membrane potential, right, because the complexes are not pumping protons. Also, mitochondrial rely on glycolysis to produce uptake more and more glucose when you treat them with doxycycline. We did it with Biology, which is a company that sells you plates with metabolites, so you can plate your cells on a dye, which is going to be reduced if the cells use these metabolites. And then we know that this dependence on glycolysis is important for survival. If you have the cells at day 6 and 9, meaning that they don't have mitochondrial function, if you change the media to galactose, that force the cells to mitochondria, the cells just die. So this is indicating that that these cells are not producing any ATP in the mitochondria. And consequently, we saw that there is a huge activation of AMPK at day. So here's the main answer to the main question: is mitochondrial important for proliferation? And it is actually important. We treat the cells with doxycycline and split them in parallel every 3 days, wild type and mutants. And as you can see, by day 9 and day 12, the differences in proliferations are huge. Also, to answer the question, is mitochondrial important for epigenetics, we did a SciLac experiment in collaboration with a group in the University of Chicago. And we saw that there is like reduction in the acetylation of a specific marks in histone 3. We confirmed this data by Western blot, and you can see, especially by day 9 of doxycycline treatment, when there is no mitochondrial function, there is also no acetylation of some of the histone marks, whereas some others don't change that much, which is more interesting because it seems that it's a specific effect. So we can say that the mitochondrial metabolism is necessary for cell proliferation and also for some specific histone acetylation. And now the question is, what function of mitochondria is driving this? I'm going to be talking today about the TCA cycle. So now the idea is we are able to destroy mitochondrial function, and now we want to restore only the TCA cycle. And how are we going to do that? Well, this is how the electron transport change works. You know this. The complex 1 and 2 are going to oxidize NADH and FADH2, and they are going to transfer the electron through the electron transport change. And when this happens, the complex 1, 3, and 4 are going to pump protons. And we are going to overexpress 2 different non-mammalian proteins. One is NDI1, which can mimic complex 1 because it has the ability to oxidize NADH. And then AOX, which can transfer the electron directly from the ubiquinol to oxygen, so kind of bypasses the need of complex 3 and complex 4. So what happens in a situation where we treat the cells with doxycycline and you don't have mitochondrial DNA, so no complexes, you are able to transfer the electrons because you have these 2 proteins. So you have again NAD and FAD in the system, so that should prevent the inhibition of the TCA cycle. So even with no complexes, the TCA cycle is going to be running again. But it's important to keep in mind that these proteins can transfer the electrons, but they don't proton pump. So the membrane potential is not being restored. So this is the situation. The TCA cycle is going to be working, so we have some metabolites back, but no membrane potential. We first confirmed that the mitochondrial DNA is being lost in these cells when we treat them with doxycycline. That shouldn't be changing. And then we did the key experiment, which is using seahorse to see the oxygen consumptions, and you can measure complex 1 activity. So these are cells non-treated with doxycycline, just normal conditions. We did it in isolated mitochondria. You basically inject the saponin, which is going to disrupt the membrane of the cells, and it's going to keep your mitochondria intact. And then when you feed them with pyruvate and malate, the substrate of complex 1, you see how there's a huge increase in the oxygen consumption. Then when you inhibit complex 1 and 3, the oxygen consumption is going to be down with rotenone and antimycin, and even more down with sham, which inhibits AOX and NDA1. It's important to see how cells overexpressing this AOX and NDA1 have higher oxygen consumption, because they are probably competing with the complexes in mitochondria. And what is very nice is when you treat the cell with doxycycline, you see how the oxygen consumptions in the control cells is very low, but the cells overexpressing AOX and NDA1 has a very decent oxygen consumption rate, which is not sensible to rotenone and antimycin. And when you inhibit AOX and NDA1, the levels drop. So complex 1 is still working in the system. We did the study of the whole metabolomic profile of these cells. You have like huge amount of data. We just filtered the data a little bit to have the pathway that we think are more important, and especially we wanted to see if the TCA cycle metabolites were being restored, and they are actually restored. When you have AOX and DA1 in the system, the TCA cycle metabolites are always higher. You see citrate, fumarate, malate, and alpha-acetoglutarate, especially if you compare day 9, you see that these levels are much higher. And the membrane potential, we confirmed that is still going down because there is no proton pumping. So we are in the situation we expected. We have some metabolites coming back to the system, but no membrane potential. And now the question is, okay, this TCA cycle restoration is driving this rescue of cell proliferation because you have some metabolites there, or histone acetylation. So for the histone acetylation, remember that mutant cells have less acetylation of histones. Well, when we do this Western blot in cells overexpressing AOX and DA1, there is no decrease in the acetylation of histones. So only having TCA cycle is enough to be able to acetylate these histone marks. But very interestingly, this doesn't have any effect in the proliferation. The cells, we treat the cells with doxycycline for 12 days, and as you can see, the rate of proliferation is the same. It's still equally decreased when you lose the mitochondrial DNA. And why is that? Probably because the cells are still very dependent on glycolysis to produce ATP. The mitochondrial is not producing ATP still. And this glycolysis is important for survival. If you change the media to galactose, the cells still die. And actually, they have an increased phosphorylation of AMPK still. So even if you have TCA cycle back, you don't have any ATP production, and AMPK is going to be activated. So this is probably repressing the proliferation. So here is where we are, or what we know is that the TCA cycle is important for histone acetylation, and that's going to be very likely important in differentiation and the differentiation of cells. And now we want to try to see we have a strategy to restore the membrane potential, even if we are losing all the complexes. And we are going to study if this is important for cell proliferation. With this, I would like to thank my lab, especially my mentor, Nav Chandel, for being such a good mentor. And also my whole lab. I think we are a very collaborative team, and that always helps. And also my collaborators, because they really make this project really easier. Thank Thank you for your attention.
Thank you. Any questions? I believe we are just beyond the schedule. It'll be nice if you could have one. Yes, sir.
Compared to cell line system, in actual tissue you characterize mitochondria and nuclear DNA and all that. So first of all, mitochondria are heterogeneous.
Yeah.
By that I mean their size is different, different genes are expressed, different proteins are transported in and out. So such models have been developed in which they are called, cells are called R0 cells. They are mitochondria deficient.
Yeah.
So first question is, why did not use instead of using a mutant and just to show that there is no mitochondria?
Yeah. So we will like about our system, you know, when people do these row zero lines, you have to, normally people treat them with ethidium bromide for like 30 days, and then you see what are the changes, right? But like this system is that system is more harmful to the cells, we would say, and also then you are like in a point when you don't have any mitochondrial function and you see which are the changes. The nice thing about our project, or our system, sorry, is that in three days you have 80% of oxygen consumption, and by day 6 you have 20%, and day 9 is basically a row zero line. So that allows you to see like the changes more gradually, and also it's only 9 days. So we think that the system is like cleaner than the regular.
So in that system, suppose you use mutant, that mitochondria also has a tendency, if you have some defective mitochondria, then you have a mixture, heterogeneous population generally. Then other mitochondria, they will start amplifying, and that will be clonal amplification.
The cells that are healthy in the system.
Yeah,
well, we took a look.
That's how it works, because one kind of mitochondria, if you suppress, then other kind of population will surpass that. So you did not observe any such.
We took a look to the morphology of mitochondria in these cells. So by day 0, they are looking like very like healthy mitochondria with the crestes and everything. When you treat the cell with doxycycline, over the days you are starting to lose like the double membrane, also like the crest morphology. By day 9, they really look like, and this is something general, we took a look to many different like cells and different parts in the cells, and they all look like really damaged. So and that's again like nice, because it's like a gradual change.
Okay, and my last thing is regarding your acetylation experiment.
Yeah.
In mitochondria in general, first of all, mitochondrial DNA does not get methylated. There are no CpG iodine. Second thing, the only evidence which I have seen, those are indirect evidence for histones. And that is only S3K4 acetylation. But physically, histones are of 4 kinds which are in general. H1, H2A, H2B, S3, like that. So those have not been reported. So do you know whether other histones have been found or not, just by using inhibitor or using immunoprecipitation method you are interpreting?
When we did the SILAC experiment, we saw minimal changes in histone 2 and histone 4. Most of the changes, for some reason, were in histone 3. We saw a little bit of decrease in the histone 4, the lysine 5, but most of the histone marks in like histone 4 and histone 2 were not changed. So that's why we focus more on histone 3, and then also to see if AOX and LIA1, when you have the TCA cycle back, you still see the acetylation of these histones. And now we are doing some ChIP-seq also to see which are really the consequences of this. This is a part that we are studying in much more detail, because like one of the explanations could be the acetyl-CoA is just limited, but we also suspect that the histone-acetyl transferase, it may be also regulated in a different way if you don't have the mitochondrial function. So that's like a very interesting point. We are there. No.
The last. Yes.
Thanks for your talk. It's great. Thanks. Quick technical question about the RNA sequencing you did on the mitochondrial transcriptome. Did you do total RNA-seq of the entire cellular content?
Yeah. So we did that in collaboration with a group. So So actually, like, I just collected the cells and sent them the samples. And yes, they did the whole RNA in the whole cell. But the analysis of all this data is going to be more for his, for their project. But they did it.
You don't know the depth of reads you had, or what was the sequence depth, or how many reads you got per sample? Is it 35 million or?
But you mean like how different?
Yeah, per sample, roughly.
Yeah,
how many?
you saw like so many differences, not only in the mitochondrial transcript but overall. Yeah. Why didn't I do it? We just did it in the whole cell. Yeah. Yeah, and we think that all this transport thing is going to be very dependent on the membrane potential. So once we have this system where we put back the membrane potential, that's something that we are going to study. Not only cell proliferation, but also see how does affect the transport of proteins in and out, and also the morphology of the cells. I bet is going to be changing too.
Yes, thank you, sir.
Thank you.
With your permission, I'm sure we'll honor Dr. Rees.
Okay, the last presentation for this symposium and this session will be given by Christine Zasada from BIMSB in Germany.
Thank you very much for the invitation and giving me the opportunity to talk about my PhD project, which is about the quantification of metabolic fluxes in cancer and stem cells to understand the metabolic reprogramming in these cells. If we want to understand a certain disease or phenotype of cancer, we have to consider the genome regulatory code. As already pointed out in several talks at these conferences, Histomarks defines which parts are accessible for the transcription machinery, so which parts of the DNA can be processed to mRNA, translated into proteins that driving the catalyzation or the conversion of metabolite A to metabolite B. Omics approaches provide the high throughput analysis of all these levels. Is the youngest one and still in its infancy, but from my point of view the most interesting one, because it combines all the above levels to one functional output, the metabolic flux. The reprogramming of metabolism is hardly debated in the last years. In 2011, Hanahan and Weinberg included the reprogramming of metabolism to their famous hallmarks of cancer. And what is occurring during tumorigenesis, a differentiated cell changed the metabolism from an oxidative mitochondrial respiratory phenotype to a glycolytic one called aerobic glycolysis, postulated by Otto Warburg in the 1930s. And what is interesting, not only this may be most famous event occurring during tumorigenesis, beside the others stated here on the CAC-MAC pathway, like the induction of hypoxia and the regulation of central carbon metabolism enzymes. The aerobic glycolysis is also a characterization of the stem cell metabolism. So during differentiation from a pluripotent cell to a differentiated cell, the metabolism is also rearranged. And what I'm even more interested in, where are the differences of the metabolism in cancer and stem cells? And maybe this provides us insights to tackle a disease like cancer. To address these questions, I combined two omics approaches, proteomics and metabolomics. Label-free and psi-like quantification by LCMS methods provide us the quantification of around 3,500 proteins, whereas a huge fraction belongs to the metabolic enzymes driving the metabolism. On the metabolomic side, I applied GCMS approach with external calibration, providing us a quantification of around 40 metabolites in absolute way, and the identification and relative quantification of these intermediates covering all over the central carbon metabolism. To move from a static to a dynamic perspective, we have to apply time-resolved measurements. For that reason, I applied 13C substrates like 13C glucose to our cells, and the time-resolved and position-specific incorporation of these stable provides us an indirect measurement of the metabolic flux. This quantitative data has to be integrated into a computational model to get real number for the metabolic fluxes occurring in a cell under certain circumstances. Therefore, I'm working together with the lab of Katharina Nö at the research center in Jülich in Germany. They had already established a non-stationary metabolic flux analysis in prokaryotic cells. And when we are talking about non-stationary metabolic flux analysis, we are looking at the cell in a metabolic steady state, so the pool sizes of metabolic intermediates are constant, and the only time-dependent component is the incorporation of the 13C carbons. Our model covers all the pathways of the central carbon metabolism contributing to the maintenance of cell proliferation and the energetic homeostasis. What we have to give as an input are the quantitative of metabolite pool sizes and uptake or secretion rates, like the uptake of glucose and secretion of lactic acid, the carbon balance in our system, and atom transitions. What we gain from the non-stationary metabolic flux analysis are hard numbers of the metabolic fluxes in backward and forward manner, and the good guess of metabolites we are not measuring by our approach. A prerequisite and a quite a technical part of my work is to match the actual fragment we are measuring by GCMS to the molecule, because of the metabolic fluxes. Here shown as an example for glutamine, we use 3 different approaches, isotope standards, because the incorporated number of carbons gives us says us how many stable labeled isotopes our fragment contains. High resolution mass spectrometry gives us an accurate mass of our fragment, and using in silico methods we have a consistency check of our mapping. So in the end we have a chemical formula and the mapping of our fragment to the molecule structure, providing to correct for the natural abundancy of isotopes in this fragment, and a correct measurement specification for our computational model. So how did I apply all these methodology to understand the metabolic reprogramming in cancer and stem cells? Together with Alessandro Prigione and his PhD student, we set up a huge cell culture experiment. We took 3 different fibroblast lines, reprogrammed them to iPS cells, and differentiated them again. In parallel, we used also human embryonic stem cells and their derived fibroblast. This gives us a possibility to compare also how similar is the metabolism of iPS and stem cells, and also their derivatives. In addition to that, as a representative for a cancer cell line, we used MDA MB231, a breast cancer cell line. To track the fate of the nutrients in the central carbon metabolism, I applied 3 different substrates: 13C glucose, glutamine, and pyruvate, because it's heavily used also in the stem cell culture medium. To match our metabolic profile with the expression of proteins, so maybe different expression of isoenzymes, we took also samples for the protein analysis. On the next 2-3 slides, I'm concentrating on the data for the human embryonic stem cell line, the derived fibroblast, and the breast cancer cell line. What is shown here on the left side in the panel is the stable isotope incorporation in percentage for glucose on the top, glutamine at the bottom, the pure, and black is always the data represented of the human embryonic stem cell, in blue the derived fibroblast, and in red the cancer cell line. And I only want to point out a few interesting facts we observed. So a cancer cell and a stem cell, they are known for their glycolytic phenotype, so we would expect a high routing of 13C glucose into lactic acid. And this is what we indeed observe. So the cancer cell in red and the stem cell in black, they have a higher incorporation of 13C glucose into that intermediate. And interesting also, this is dropping after the differentiation. Here shown for 2 stem cell lines, we used H1 and H9. Another interesting finding was the routing of 13C glucose into the metabolite serine. Serine connects the central carbon metabolism with the one carbon, with epigenetics representing the entry to the one carbon metabolism. So this pathway providing the precursors, for instance, for methylation of histones. And also here we observed a drop of 13C glucose routing after differentiation. Interestingly, we don't see any incorporation of 13C glucose into that intermediate in the breast cancer cell line, giving us a distinct feature of cancer and stem cell metabolism. So glycolytic phenotype of the cancer and stem cell. What is about the fibroblasts? They're known for mitochondrial, so oxidative phosphorylation, and indeed we observed an increased routing of 13C glucose into TCA cycle intermediates, here shown in the bottom for FIT. Also the breast cancer cell line shuttles a lot of the glucose into these intermediates and run in that way the TCA cycle. What I want to point out with that slide is the huge advantage of GCMS measurement. The carbon resolved and position-specific analysis gives us deep insights how hubs are working in the central carbon metabolism for the pyruvic acid. This metabolite is really interesting because it connects the glycolysis and the TCA cycle. And depending on enzyme activity, we can observe a different number of labeled carbons incorporated into TCA cycle intermediates. The shuttling from pyruvate via the pyruvate carboxylase to oxidase gives an incorporation of 3 stable isotope labeled nucleic acid. The conversion of pyruvate by the pyruvate dehydrogenase over acetyl-CoA to citric acid gain in 2 stable labeled carbons into citric acid. Due to the cycling and the symmetry of our intermediates, we end up then with 4 or either 2 at the bottom or at the top labeled 13C carbons. And this is shown here in this panel. So we have malic acid, the incorporation with 2 carbons, 3 carbons, and 4 carbons. And for the differentiated fibroblasts, we see that both enzymes, the PC and PDH, are working much higher activities than the cancer and the stem cell line. But this is what I also want to point out here. This is a good example why we need computational approaches. Just by eye and hand, we are not anymore able to interfere from this data, the metabolic fluxes and what's going on at this complicated system of TCA cycle. What I want to further point out is that we don't see such a huge difference in the routing of 13C pyruvate into lactic acid. Remember, we have a higher routing of 13C glucose into lactic acid for the stem and breast, for the stem and tumor cells, but also the alanine synthesis coming from the pyruvate is increasing after differentiation and the difference to human embryonic stem cells and the cancer cell line. I already want to summarize, and I hope that I convinced you that the application of 13C stuff is absolute computational method is a good tool for metabolic reprogramming in rather here in the middle summarizes the fate of 13C glucose into 5 key intermediates of the central carbon metabolism. I showed you data confirming the glycolytic phenotype of stem and cancer cell here shown with a higher with the cycle by glucose into TCA intermediates. With that, I want to thank you for your attention. My supervisor, Stefan Kemper, all the great lab mates and my cooperation partners, and I'm happy to take your questions.
Any questions?
Very nice talk. So my question is, when you use carbon-13 labeled pyruvate and look at the flux, the carbon-13 migrated to the product of different locations. If migrated to one position, how do you know, based on the one mass difference, based on the mass spectrometry peaks, where it is located? If you have two, they have some random distributions. For this kind of isomers, how the mass spectrometry peaks differentiated between the different isomers where the carbon-13 is located?
So therefore you need the computational, meaning the atom transitions into that network. And there's a complicated mathematical framework Katharina Nö and Wolfgang Wiechert established. There are several papers out of it, and it's hard to explain just in one sentence, but they take into account these different isomers and calculating the metabolic flux. So that's really the reason why you need the computational methods in the end to understand that, because it can just measure the shift of intensity depending on the number of incorporated isotopes, and then you have to calculate, and the computer gives you the answer how the fluxes are going on in the end.
Yeah, just one suggestion to you: if you also incorporate MR metabolomics for carbon-13 MR, because carbon-13 is isotope, and actually you are able to show different peak positions because they have different chemical shifts. Something you may consider.
Sorry, I didn't get.
For MR metabolomics.
NMR.
Yeah,
okay.
using carbon-13 MR. So because different locations
okay.
of the carbon-13 in the molecule, they show different peak positions.
Yeah. Okay.
Yeah, that's one thing. So do you have a graph to show the flux changes as a function of time?
Yes, this is currently ongoing work. So at the moment, the computers are calculating the metabolic flux maps, and unfortunately, I couldn't present the data because hopefully in the next 2 weeks I have these kind of data to match this with the proteome, which I also didn't show this time, with the metabolic profile to get further insights into that, to get the complete picture out of this.
Since you showed data about pluripotent cells, so one big problem in cancer field is either you have iPS cells, or you have cancerous cells, or you have other kinds of cells. Yeah. But what we want, that out of all stem cells, out of 1 million cells, only 1 or 2 cells are cancer stem cells. But yeah, there are some markers, receptor markers, by which you can enrich or isolate. So are you planning, or has somebody done metabolomic analysis of cancer stem cells?
I'm hardly discussing this topic with Stefan all the time, because it always, if you look at stem cells and cancer cells, why you have these cancer stem cells. The problem with cancer stem cells is, so you need for proteome analysis maybe 500,000, 1 million cells to do proteomics. For metabolomics, GCMS, I don't know how it is with NMR, you need somehow 2 million cells. So, and what I think what the drawback, you have to purify these cells to get so many cells and do this stuff. I think this is now the limitation at the moment for us to go in that direction. But this is something, after I wrote my thesis, I think, and I spent some more time in Stefan's lab, I'm going to do that, to see if there are further differences, or where do they fit into these different parts.
Because if that problem can be solved, we can get more number of cancer stem cells, then many things can be done, and those can be targeted.
Yeah.
Very good.
Very nice talk, and I think it's a very powerful method. You should be able to do this also keeping the cells in hypoxia, right?
Yeah.
And then just, because I think it could be very interesting, especially for the sample sharing stuff that you were presenting in cancer. this one carbon metabolism has been shown very recently to be especially important in hypoxia to keep the redox balance. And at the end, I think so we normally make statements when we study this in cell line, and it's basically the tools we have, but the reality is that the tumors are very heterogeneous, and the hypoxic conditions are not the same in all the cell lines. So I was just curious to see if you could do it in hypoxia.
So we have this hypoxia chamber, and we are working with cancer cell lines under different oxic conditions, also changing glucose level and all these things. This is also something I have in my mind for later, to take IPS and all these cells and put them under hypoxia to see how they behaving. In the end, it's about in vivo, in vitro, right? You have this environment, and it's hardly to get this real working in cell culture experiments. But yeah, it would be just technically nice to see what is changing on the level of metabolome and proteome under lower oxidant
Okay, thank you. If no other question, we'll honor this one. Thank you. Wonderful presentation. Thanks.
Sorry, I have a last question. Please.
Okay.
Sorry.
This will conclude.
I have last question.
Oh, you have a question? Yeah.
I have a last question. On the proteomic data, do you interrogate the proteomic data in the computation of the fluxes, or do you integrated the proteomic data to constrain the computation of your fluxes? Okay. I want to know if you integrated the proteomic data in the computation of your fluxes, or you only use this after the computation of the fluxes. Yeah. Yeah. Yeah, because the protein can regulate, yes, the transformation of metabolite. Okay, thank you very much.
Okay, this will conclude the Metabolomics 2015 conference. Please join me to thank all the speakers for this session, and then I hope to see you again in another conference in this area. Okay, yeah. See you then.
We're happy to invite you to the next conference probably in Osaka, Japan, 2016. Bye. Okay, thank you.